So regarding the homeosis, we have the ultrabithorax and the antennapedia. Ultrabithorax, what does it mean, ultrabithorax? So here we have a Hox gene mutation that will induce in flies the formation of, as we can see here in this figure, a development of a second thorax. It's like the fly is having two thorax instead of one thorax, okay, so that we can get, after the mutation of the gene UBX, the third thoracic segment. Okay, usually we have on it two rudimentary structures attached called halter. Halters are very rudimentary structures that could not be developed into wings like what it happened on the second thoracic segment, since the wings are attached on the second thoracic segment and not on the third thoracic segment. So instead of these halters present or attached on the third thoracic segment, the mutation of the UBX, which is one homeotic gene, induced to the formation of wings instead of halters, even on the third thoracic segment. And this will make the fly like having two thorax instead of one thorax. Okay. So these flies are called four-winged flies instead of two flies. And of course, this is not very healthy for the fly. Okay, this will not be very helpful for the flight. Okay, so here we can see the halters are always attached to the third thoracic segment in a normal fly, while the wings are present and they developed usually on the second thoracic segment. When we make this, when we have a loss of UBX, ultrabithorax, this will develop wings as well on the third thoracic segment instead of halters. Okay, so that's why we get four-winged flies. Regarding the second homeotic mutation, it's called antennapedia. So here, for example, by electron microscopy, we can see a wild-type Drosophila head, and you see this is the place of, this is the localization of the antenna. Okay, these are the antenna usually attached to the proboscis. Okay, you see the antenna, while in antennapedia mutation, when we have a mutation of this gene called antennapedia, we have a development of pedia, it means legs, instead of antenna, and that's why this gene is called antennapedia, because it will give rise to the development of pedia, or legs, instead of antenna. Okay, I remind you always, in Drosophila we call the gene according to its phenotype. Here we got legs instead of antenna, so they called it antennapedia. So now we'll talk about the specification of the anterior posterior axis in Drosophila, and especially this will happen very early during oogenesis, even before the release of the oocyte and its fertilization. So how this anterior posterior axis of the Drosophila embryo is established during oogenesis. So if we take one egg chamber, as you can see, each egg chamber is formed, of course the mature egg chamber is formed by one oocyte and by like 15 nurse cells surrounding them. So one oocyte, 15 nurse cells, and these are surrounded by follicle epithelial cells to protect these oocyte and the nurse cells. So nurse cells, they are called like this since they provide the nutrients. They work like a nurse, okay, to provide nutrients and the different messenger RNAs important for the development and for organizing oocyte body axis. So during the establishment of the anterior posterior axis of the oocyte, we have the oocyte will move into the posterior region. Okay, the oocytes nucleus, okay, will move towards the posterior region of the egg chamber, okay. So as you can see, once the nucleus is just present towards the terminal follicle cells, so they are called terminal follicle cells, they are not called posterior, since the anterior posterior body axis is not yet established. The nucleus will just approach to these terminal cells, and by this time the nucleus will produce one protein, okay. This protein is called gherkin. So we have gherkin protein released. Gherkin will bind specifically to one receptor present at the surface of the terminal follicle cells. You see, these are the terminal follicle cells. So that we have gherkin will find torpedo. Torpedo is the gherkin receptor, and torpedo is present at the surface of those epithelial or terminal follicle epithelial cells. So when gherkin binds to torpedo, these terminal follicle cells will be differentiated into posterior follicle cells, and then these posterior follicle cells in purple here, they become able to produce one kinase, which is the protein kinase A, or the pKa. This protein kinase A is essential for the orientation of the mitotic spindle, okay, the orientation of the microtubules of the mitotic spindle, that will happen later, as you can see here. So that the growing ends of these microtubules will be located at the posterior end, while the non-growing end, the minus ends, will be present at the future opposite side, that will become anterior. So now the nurse cells will provide the cytoplasm of the oocyte with specific maternal messenger RNAs. With messenger RNAs, these messenger RNAs will bind to carriers present on these microtubules, okay, and these carriers will just carry up, okay, these messenger RNAs toward the plus end, which is the posterior pole, or the opposite end, that will become the anterior pole. Let's talk about bicoid. Bicoid messenger RNA is synthesized by the nurse cells, and then it's released into the cytoplasm. It will pass to arrive to the cytoplasm right here, to the cytoplasm of the oocyte. And you can see here, after the formation of these microtubules, okay, we have an elongation of the oocyte. So the size of the oocyte becomes more elongated than before, okay, due to this elongation of these microtubules. So bicoid, when it binds to dynein matter present on these polarized microtubules, this dynein will carry this bicoid messenger RNA into the non-growing end, okay, to be localized at this position, okay, so that we have a concentration of bicoid messenger RNAs at this location. While concerning Nanos and Oscar, okay, Nanos and Oscar are also released and produced by the nurse cells. Once in the cytoplasm of the oocyte, they will bind to the kinesin 1 motor, okay, to the kinesin 1 motor protein, which is a carrier that will carry Oscar and Nanos towards the posterior or the plus end of these microtubules, that will become the future posterior end after the localization of Nanos and Oscar, that will be associated right here to determine the posterior pole. So once bicoid messenger RNA is located right here at the anterior, this part will be called anterior, while the opposite domain after the localization of Nanos and Oscar, it will be called the posterior pole of the oocyte. So this way, the anterior posterior body axis of the oocyte will be established. Okay, so this is the way the anterior posterior axis is established. And after the establishment of the anterior posterior body axis, another axis also will be established, which is the dorsal ventral axis. That's why you see that the nucleus, okay, will not be located anymore here near the posterior side, okay, since the anterior posterior axis is already established. So the nucleus will be moving from this side, the posterior side, to this location in order to establish the dorsal ventral axis. So regarding the anterior posterior location, they did an in situ hybridization of an egg chamber. You see, this is the oocyte. These are the nurse cells right here, and these are the surrounding follicle protective cells. So this in situ hybridization was against bicoid messenger RNA to see where bicoid is localized, and you see that the messenger RNA of bicoid is only localized at the anterior pole of the oocyte. Okay, so it's a main determinant of the anterior pole of the oocyte. And when they did the opposite in situ hybridization for Nanos and Oscar, they also only found them localized at the posterior end of the oocyte, but nothing in the anterior, since they are only specific for the posterior end. So here we talked about the messenger RNA of bicoid. Okay, so this messenger RNA will not be translated into protein before fertilization. Once the oocyte is fertilized, the maternal messenger RNA of bicoid will be translated into bicoid protein. And bicoid protein is a diffusible protein. So you see it can diffuse from the most anterior end of this embryo, okay, to the other less anterior structures, okay, but all of these parts are still anterior. You see, nothing is expressed at the posterior part or in the middle, okay, but we see that we have a gradient of bicoid expression in embryos, okay. We cannot, because you know that the protein can diffuse but not the messenger RNA, okay. So in the oocyte we only have an accumulation of bicoid right here. We don't have any diffusion of this messenger RNA. After fertilization, messenger RNA is translated into bicoid protein, okay, so that this protein will be able to diffuse. Okay, we see a gradient of bicoid expression in the wild type, and if we compare this wild type expression of bicoid in the so we have the highest levels are at the most anterior side, and as far as we move from the anterior pole, we have less expression of bicoid, to be null by the posterior end. We don't see any bicoid expression. Okay, you see this graph in black. But if we compare this graph with the expression of bicoid in a bicoid mutant embryo, we see that in the bicoid mutant we don't have any expression of bicoid, even or all over the anterior posterior body axis.